Well, thank you very much indeed for being here today. Let me start by thanking Joanne for her truly inspirational leadership. It's been a pleasure to have known you, Joanne, over many years, and you've truly led the integration between care and compassion, which is so important as we look forward. You know, it's interesting that today in medicine, we talk about the importance of interdisciplinary team-based care. We talk about the value of quality, of the connection of the caring physician and other providers, of the value of translational research that brings basic discovery from the laboratory to the clinic and back. We talk about the importance of continuity over the life journey, and as we've heard already, of combining compassion with care as we proceed. And while we look forward, it's important to perhaps just look back and remind ourselves that pediatric oncology has really been the exemplar of all of these issues. I guess at this juncture in my sort of personal life journey, I'm old enough to have been around almost at the kind of beginning or early beginnings when, as a medical student, I well remember, like many of you, seeing my very first patient. In my case, it was a young child with acute leukemia, and it was a devastating time. That was, quite honestly, in the late 1960s, and most children died. And I made a resolution then, which is that I would never think about going into a field like pediatric oncology. I couldn't imagine how anybody could do it. And then years later, by a matter of happenstance and coincidence, that happened. And I had the privilege of starting a journey, as many of you did, in the field of pediatric oncology with colleagues, including Greg, who is one of the early fellows, not far behind me, I would say. And we watched pediatric cancer go from incurable to treatable and curable. And then we watched, as we well know, Smita, you're going to speak to, all of the complications that took place. So that the early discoveries, while having a dividend, had unanticipated complications. But we also learned from the very beginning the importance of integrated care, of team-based care. I see my colleague, Lori Weiner here, who really led much of that effort, certainly in my world, of bringing together the care team in a coordinated and integrated way. We watched not only basic discovery and translation lead to new opportunities, but to the importance of looking forward to further defining the future of both pediatrics and indeed all of oncology. And Otis and Pat, I would say that pediatric cancer has certainly stood, even with its rarity, as the underpinning of most of modern oncology as we see it today, and hopefully as we look forward to the future. So with that as sort of contextual background, looking forward requires us to continue to build on this effort. I think Joanne also spoke to the importance of keeping our minds on the connection between what we do in research, how we bring that into the care of patients, how we continue to engage parents and families as voices that remind us of the importance of moving forward on their behalf, on behalf of their children. That's the spirit I think that we come here today with. And without further ado, and Greg, you've already been given the mandate, which I won't repeat, except I will cut you off. So, Greg Reaman.
Well, thank you very much. It's an honor and a privilege to be here and to be part of this. And I hear your admonishment, Joanne, and I will, but I don't know how to advance the slides here. So, so don't start timing me yet. So I'll start with disclosures and disclaimers. As a federal employee, I have no disclosures and no financial interests. I won't spend a great deal of time on the progress in childhood cancer. I think you have heard about it. I do want to just point out that I think One of the pivotal reasons for this success is the unique integration of clinical practice and patient management and clinical research, and the fact that we do have a highly effective national clinical trials infrastructure. As we talk about therapeutics and new treatments, I think it's important to point out that we have always highly leveraged adult drug discovery and development. I think there's an opportunity to change that somewhat, but I think we're going to continue to have to leverage adult discovery. So despite the progress, childhood cancer remains a problem. There are still nearly 2,000 children who continue to die each year. Advances are not seen and have not been seen for some diseases and stages of disease. And has already been said, but I don't think we can say it enough, is that for even those children who do survive, reduced quality of life due to cancer, and more importantly, its treatment, is something for which we remain responsible. So childhood cancer mortality varies by age groups. In younger children, brain tumors and acute leukemias are the primary reasons for childhood cancer mortality. That changes somewhat in adolescents, older adolescents particularly, again with brain tumors, but with bone and soft tissue sarcomas also playing a major role. So I think there are some real challenges to therapeutic research, and specifically therapeutic research that is aimed at drug development. They're both pragmatic and scientific. The low incidence of the disease, fortunately, there are only 15,000 patients per year. We have small study populations for clinical trials. There are significant pharmacology considerations and effective normal development and maturation on organ function and metabolic enzymes, so it makes it somewhat difficult. And formulation requirements for pediatric use. So for parenteral therapy, that's not an issue, but most new drugs are really now being developed as oral formulations. So giving a capsule that would choke a horse to a 2-year-old child is not possible. So asking companies to develop pediatric appropriate formulations is something that is in fact required. The current industry research and development models clearly aren't favorable to pediatric drug development. With 15,000 patients totally per year, there is no market, if you will, for drug development and marketing. We have some excellent preclinical model systems and preclinical testing programs. Despite their excellence, they're insufficient. They're not inefficient, but they're insufficient. There's not enough funding to support the efforts that are clearly needed in this area. The long-term availability of promising products, those products which are developed primarily for adult cancers, but for which there could be pediatric relevance, which if they fail in adult testing, disappear. And unfortunately, that deprives us the opportunity to test what might be a promising drug in the pediatric population. There are clearly very different perspectives on clinical benefit, and at the FDA, it's all about clinical benefit, weighing benefit and risk. And benefit in the adult cancer population is prolonging survival or prolonging progression of disease by 2 months, 3 months. In pediatric oncology, we're not interested in prolonging survival. We're only interested, or primarily interested, in cure and quality of life. So approaching cancer as a chronic disease is really not and never will be a pediatric perspective. The advances in cancer biology, I think, have been unprecedented with respect to genomic medicine and providing the molecular pathogenesis of cancer. Genetic mutations and amplifications involving major signaling pathways have emerged as validated targets for drug development. And I list some examples here, imatinib or Gleevec in CML and Philadelphia chromosome-positive ALL, probably the most notorious and certainly the first of a series of, I think, what we're going to be seeing. But all of these that I list here, crizotinib in non-small cell lung cancer, or at least a small subset of non-small cell lung cancer, vemurafenib, BRAF inhibitor in melanoma, all have potential implications for pediatric oncology. And it's imperative to follow through on those potential implications. So of the multiple molecularly targeted therapies, new molecular entities as of last week, there's actually 54 now, approved for advanced stage or recalcitrant adult cancers, based on, again, on a favorable benefit-risk consideration. None of these have yet demonstrated to be curative, at least as single agents, and the whole field of personalized or precision medicine has really largely benefited adults. Molecular drivers of cancers are not universal across specific histologic types, and there are multiple orphan designations, so small subpopulations of what were common adult cancers. And these genomic abnormalities are not unique to specific histologic types, and therefore multiple biomarkers may exist for the same cancers, giving rise to molecular characterization, and the NCI MATCH trial is an example. The vast majority of recurring genomic alterations for childhood cancers have been identified. This is really significant team science efforts led by the NCI TARGET, Therapeutically Appropriate Research to Generate Effective Therapies Initiative, and the St. Jude Washington University Pediatric Cancer Genome Project. But alone, Target has sequenced over 1,200 cases using whole genome sequencing, and sequenced nearly 3,000 additional cases with frequency validation. So what we've learned, childhood cancers have fewer gene mutations than adult cancers. That doesn't mean that there aren't still opportunities for precision medicine and the utilization of genomics in pediatric oncology. Many childhood cancers are driven by mutations that are either rare or don't even exist in adult cancers. Histone mutations for diffuse intrinsic pontine gliomas and high-grade gliomas, and some very distinct and unique fusion genes in sarcomas and some brain tumors. And most childhood cancers at diagnosis, because of the embryonal nature of their origin, lack mutations in genes that are relevant to those approved molecularly targeted agents or for those that are being developed for adult cancers. Fortunately, the relatively small number of patients with any specific cancer does create challenges in conducting trials to reliably identify effective treatments. It's complicated further by subdividing diagnoses based on genomic characteristics. I think one excellent example is medulloblastoma, but even before medulloblastoma, acute lymphoblastic leukemia, the most common childhood cancer, is now not one disease. We've known that for years, but every several months we know that there's a new subclass of ALL. And the other thing is that many of these genomic alterations only occur in a relatively small percentage. So in high-grade gliomas with BRAF V600E mutations, only 5 to 10% of these tumors actually have the mutation. So medulloblastoma now appears based on genomic characteristics to actually be 4 different diseases with different clinical features, different presentations, different histology, different survival rates, and can some of these genomic features actually be used to define and develop unique and specific therapies. Multiple challenges, obviously. Challenges, I think, can become opportunities. There really aren't any opportunities for prevention and early detection in pediatric cancer, unlike adult malignancies. There are relatively few driver mutations present at diagnosis that are addressed by currently available targeted agents. And the known driver mutations of pediatric tumors like EWS-FLI1 and Ewing sarcoma, Pax-4-Ked, and alveolar rhabdomyosarcoma, there is no specific interest in the part of pharmaceutical companies to actually look at targeting these abnormalities. So again, we have to look at addressing the repurposing, if you will, or the secondary indications of targeted drug therapy for those agents that are being developed for adult cancers. I think there are opportunities to maximally utilize the regulatory authority that's provided by current legislative initiatives to address indication-based waivers. We have two pieces of legislation, one of which mandates evaluation of drugs in children if the indication is the same. But children don't get breast cancer and lung cancer, prostate cancer. But there are opportunities to provide incentives for studies if there's appropriate biologic and scientific rationale to do so. We need to rationally prioritize those products to be evaluated. We have to redesign early phase studies looking at toxicity and dose selection. So the current history or the current sequence of phase 1, phase 2 studies really has to be reevaluated and rethought. And there really has to be early communication and collaboration between investigators, patients and families, industry, and regulators. I think the other big challenge and the major challenge, the bigger challenge, is really new drug development when no adult indication exists. Currently, there is no legislative fix. I think the legislation could be addressed by adding maybe more substantial and early incentives to industry, because the current incentives actually come at the end of a drug's life cycle, and providing earlier incentives may de-risk or provide a de-risk to companies for early development. I think the continued success of current special initiatives like the Pediatric Rare Disease Priority Review Voucher is subject to dilution of benefit in competing priority review mechanisms. But again, here, the early development incentive is lacking. I think there's an excellent example of public-private partnerships in the role of the NCI with dinutuximab, the chimeric 1418 antibody in neuroblastoma. But I remind you that the time to actually realize this was almost 2 decades. AMG 479 in Ewing sarcoma is another. So I think the role and the importance of public-private partnerships is real. But we have to do something about the timing. There continue to be opportunities for precision medicine in pediatric cancer. The tyrosine kinase inhibitors in Philadelphia chromosome-like, not pH positive, but other genomically defined subgroups of high-risk ALL. A pediatric MATCH study that is going to look at the molecular analysis of tumors for Therapeutic Choice. And there are already genomically-selected targeted agent approaches under development in neuroblastoma, medulloblastoma, and AML. I think another area that really requires exploration more than is currently happening is in the approach of immunity with monoclonal antibodies, immunoconjugates, enhancing host effector T cell function through checkpoint inhibitors, and now more recently engineered cell therapy. So with monoclonal antibody, we have the example of combined immunotherapy with cis-retinoic acid in improving the outcome for children with high-risk neuroblastoma. And that is actually proceeding, and a biologic licensing application has been submitted to the FDA for approval of this agent. Blinatumomab utilizes a unique BiTE technology, or bispecific T-cell engager, with an antibody that addresses the CD3 antigen on T lymphocytes, cytotoxic T lymphocytes, and the CD19 antigen on B precursor ALL cells, causing a temporary synapse and inducing T-cell response to kill tumor cells. The role of checkpoint inhibitors, the immune system has a natural sort of braking mechanism to prevent autoimmunity, both through the CTLA-4 antigen on T cells and the PD-1 antigen, which interacts with PD-L1 on tumors in inhibiting either the PD-1 antigen on T cells or the ligand on tumor cells. Has actually been demonstrated to have pronounced effects in a number of pediatric, or in a number of adult cancers, most notably melanoma. The development of CAR T cells, or chimeric antigen receptor cells, I think holds a great deal of promise already in ALL. And this is just a cartoon demonstrating a T cell that recognizes the CD19 antigen that's present on every B cell ALL. And causing lysis. High rates of remission induction in children with ALL treated have been demonstrated in a series of about 39 patients, and this is information presented at this year's ASH meeting by Steve Grupp from CHOP. A 92% reinduction rate with actually prolonged duration of remission, some up to over 31 months. And for those patients that have continued for a year, no events after a year. I think the other thing that we have to be concerned about with the potential for new therapeutic interventions are the short-term and long-term safety concerns. This is going to create continuing challenges for long-term longitudinal observation. Despite the promise, targeted cancer agents are not non-toxic. So all we've heard about is targeted therapy, more effective, precision, no toxicities. Nothing could be further from the truth. They're just very different toxicities. These have unique effects on target tissues, which depend on pathways being inhibited, and there's definitely a heightened potential implications for children, given the developmental considerations. So if you look at the list, they're very similar to what we see with conventional chemotherapy, but here the mechanisms are frequently not completely understood, but are very likely very, very different than those induced by cytotoxic agents. And this is particularly true for those checkpoint inhibitors, where autoimmunity is a major problem that really needs to be considered. So we don't have sufficient long-term exposure data in adults to really advise us on how best to proceed in pediatrics. There's little combination toxicity data, and it's unlikely that any single drug is going to be used as a single agent in pediatric cancer. You know that we have a history of developing the whole field of multi-agent chemotherapy. That's not going to change. So integrating targeted therapy with more standard regimens is a reality. I think the organ system targets and the surveillance intervals are going to have to be reevaluated as we go forward in new clinical trials. And this will really be a new paradigm for pediatric cancer long-term follow-up. I think one good thing is that there are really increased opportunities for industry collaboration to meet the requirements for long-term registry studies, which the agency will require. And this could actually provide some very important support for long-term follow-up studies in pediatric oncology patients. Patient-focused drug development, the agency is committed to this. Is it a reality for children? At the current time, I don't think so. But incorporating patient experience perspective in changing, not necessarily changing, but just realizing and defining what is benefit and risk. The use of patient-reported outcomes or observer-reported outcomes as evidentiary standards to support treatment benefit claims. And we're talking about the important concepts of interest, survival, symptoms, and function. There are challenges with the development of PRO instruments in children with cancer, but again, we're aware of those. We've sought some input from the International Society of Pharmacoeconomics and Outcomes Research at a recent pediatric subcommittee of the Oncology Drugs Advisory Committee. The problems are comprehension, content validity, and domains of interest. I know it's blinking. But there is a potential role for combined PRO and observer-reported outcomes. There is some limited experience with qualification by the agency's SEAL team in plexiform neurofibromas. I think we can learn from that as we seek to look at potential PROs as endpoints. There's limited success in oncology in general with these PROs as endpoints because of the effect of concomitant meds, the requirement for blinded and randomized trials, which again will be difficult in children. They need to be objective and quantifiable measures. So I see going forward that pediatric oncology is not the glass half empty, but really the glass half full. I think there are expanded opportunities for evaluating precision medicine approaches to improve outcomes and to utilize those measures early. It will necessitate paradigm shifts in study design, when studies are initiated, and how they're conducted and follow-up. There really needs to be a rational science-based strategy for prioritizing which and when new products should be tested and in what diseases, and how these could be successfully integrated with standard therapy. And there really needs to be expanded collaboration between patients and families, investigators, industry, and regulatory agencies. And with that, I will stop. Thanks.
Thank you, Greg.